Welcome to our channel Bio by Gulab Sir. As a part of series of lectures, today's topic is isolation of DNA fragment or isolation and separation of DNA fragment. As we have studied different topics under biotechnology head, biotechnology principles and processes. This is also one of the process that isolation and separation of DNA fragment. As you know that DNA is negatively charged. We required isolation of that DNA for various purposes, for DNA fingerprinting and for DNA recombinant technology, to transfer gene of our interest into bacteria through vector plasmid. So we need to get them separated from the source. So we have technology, gel electrophoresis is the machine that helps to separate DNA fragment from rest of the other DNA, desired DNA fragment from rest of the other unwanted DNA fragment. So see, the construction of that machine is gel electrophoresis machine. Gel electrophoresis. It is so called because here we use agarose gel. Agarose gel. This agarose gel is a natural polymer. What is it? It is a natural polymer obtained from algae, Xylidium and Gracilaria. This natural polymer obtained from algae, Xylidium and Gracilaria, obtained from, for NEET point of view, obtained from Xylidium and Gracilaria, algae. So remember that word, Xylidium and Gracilaria. Here it work on two principles. This work on two principles. One, DNA is negatively charged. Negatively charged. And being negatively charged, when it is added towards the cathode end, it moves towards the anode end. Second principle, size of the DNA. Size of the DNA fragment. Smaller one, length wise, if it is more in length, move slower, less in length, move faster. So larger one or longer one, longer fragment moves slower, while shorter moves faster. These are the two principles help to separate that DNA fragment from each other. Now here we have this machine, gel electrophoresis. See how it is being. Here it is rectangular, a box, glass, made up of glass. Here there are wells. Here there are wells. Depend 3, 4, how much we required so these are the indentation. From here we can put fragment of DNA and it is filled with gel electrophoresis. This entire box is filled with agarose gel, and one end of this one, that is cathode and another is anode, and they are connected to the electric, either through stabilizer. It is connected through stabilizer. Control amount of current will be passed. So this is what, suppose a box where that key switch is also there to switch on and off. So only one thing we have to remember, This end is called cathode, is a positively charged, and this negatively charged, and this one is called anode, is a positively charged. Now when we put the DNA fragment into the well, they will keep on moving to the direction of anode. So this is the direction of movement. These areas called well, this is cathode, this is anode, and it is filled with agarose gel, filled with agarose gel. Now this DNA fragment which move, so as we have written here, that longer one moves slower and shorter one moves faster. So naturally longer one are behind and shorter one they are ahead. So see, those are which are very short, they move faster and they are separated from longer one. It is because of that electric charge which apply on that negatively charged fragment with the size enabling them being shorter move faster, and therefore they reach early to the anode end than that of longer one. And therefore I wrote here, these are the two principles, that DNA is negatively charged, and therefore when we placed this DNA fragment into the well on the side of cathode, so that is negatively charged, so opposite charges attract, and thereby this negatively charged DNA moves towards the anode end, and thus they get separated from each other on the basis of their size, that longer one moves slower and shorter one moves faster. Now who are that shorter one? They are digested DNA fragment by restriction endonuclease with specific set of palindrome, or specific DNA fragment is cut by specific restriction endonuclease, and thereby they are recognized. Undigested one, they remain behind because they are longer one. Therefore we can write here, these are longer and undigested, and these are digested, and it is written very clearly wherever you search in net or in book, it is very clearly written that they are digested because they are cut by specific restriction endonuclease enzyme. Now see, when these fragments are separated, now they are stained by, next step, that they are stained by ethidium bromide. So this is the stain is used to stain these fragment, ethidium bromide. This is brown color, brown in color, that stain is brown in color, so that this fragment appear brown in color, but they do not appear in white light or in the wavelength between 400 to 700 nanometer. They are exposed to the UV radiation. So stained by and followed by exposure to UV rays or UV radiations. So this help to see. So staining and exposure to UV radiation help to see that fragment present inside the gel electrophoresis machine. So as they are appear brown in color against UV radiation, they can be cut and separated. So next step is elution, cutting and separation of DNA fragment, desired DNA fragment is known as elution. Remember this word elution, as it very often asks what is elution. It is the cutting and separation of DNA fragment from gel electrophoresis machine. Now this cut piece of DNA are ready. What is the purpose? Purpose is this cut piece of DNA, pieces of DNA or fragment of DNA are used For one, DNA fingerprinting, and second, DNA recombinant technology. DNA recombinant technology to obtain the specific product from that desired gene. Say, for example, gene of insulin that we can separate, and it can be ligated with the vector to introduce into the bacteria to obtain the product of that gene, as that gene can express, that is undergo transcription and translation to produce specific protein in bacteria. So from bacteria we can extract the product of that desired gene, expected product of that desired gene. So that fragmented DNA are isolated and separated for these two purposes, DNA fingerprinting and for DNA recombinant technology, to recombine it with plasmid and transfer into bacteria through vector to obtain its product. So once again, gel electrophoresis is a technique to have the separation of DNA fragment, isolation and separation of DNA fragment. So very purpose is to isolate and separate DNA fragment. So here, to isolate and separate DNA fragment, isolate and separate DNA fragment. I hope it is very clear to you that how this gel electrophoresis work to have the isolation and separation of DNA fragment on the basis of these two principles. As DNA being negatively charged, very important that which part of the DNA that is negatively charged so as that get attracted towards anode is very important. Phosphodiester bond that form backbone of the, once again very important, phosphodiester bond. So I mention here in bracket to understand that phosphodiester bond, exposure of that phosphate with that phosphodiester bond is negatively charged, and that provide overall negative charge to the DNA. So as a part of NEET question can be asked, which part of the DNA provide negative charge to the overall DNA fragment to have the isolation and separation on the basis of that charge being negatively charged move towards the anode or positively charged N. So phosphodiester bond and phosphate being present inside the backbone of that DNA helix structure is negatively charged. I hope you did understand the very mechanism of isolation and separation of this DNA fragment with the help of gel electrophoresis machine. Still, if you are in doubt, you can ask in comment section. I appeal you to like our channel by subscribing it, and by pressing bell icon you will be notified time to time when new videos will be uploaded. Thank you.